Hello everyone. So today we are going to start a new chapter, acid, bases and salt. This chapter comes under your chemistry section. So let's get started. So today what we are going to learn, today we will learn a little introduction about these chemicals, acid, bases and salt, and then we will learn about indicators for testing acids and bases. What are the indicators we will learn over here? Litmus, CHINAROSE indicator, turmeric indicator that comes under natural indicator, and phenolphthalein indicator which comes under synthetic indicator. So in brief we will discuss about these indicators and a little introduction. Now this acid, bases and salt, these are the three important groups of chemical compounds, or you can say chemical substances, which are useful to us in many ways. For example, in our previous chapter what we have learned that our stomach makes an acid known as hydrochloric acid, which is necessary for what? Digestion of food. Okay. Now baking soda, the chemical name is sodium hydrogen carbonate. It is used for baking bread. It is a kind of base. Whereas common salt, that is sodium chloride, it is used in cooking food as a salt. Okay. Now some of the acids and bases and salts occur in nature, whereas some of the acid, bases and salt, they are made artificially in factories. Now there are however many other chemical compounds, okay, which are neither acids nor bases or nor salts. They have got the pH 7. Those are known as what? Those are known as neutral substances. If we consider a pH scale, okay, pH scale, now what happens? This pH scale, it ranges from 1 to 14. Okay, now 7 is the mean pH scale. Now below 7, what we consider below 7, it is the acidic, okay, and above 7 it is a basic, and 7 we consider it as a neutral. So certain substances like we can say pure water or distilled water, glucose, cane sugar, urea, then common salt, they are neither acid nor bases. Okay. Now in this chapter we will study only with acid, bases and salt. Okay. Now in order to find whether a substance is an acid or base, we should first know the meaning of the term acid base indicator or just the indicator. Now what are indicators? Indicators, these are the special type of chemicals which are used to test whether a substance, okay, or whether a given substance is what is an acid or a base. Okay, and what is its function? That means how you can find indicators. It changes their color when added to a solution containing acid or a base. Now the next topic is indicators for testing acid and bases. Now what is an indicator? Here you see an indicator is a dye. Dye, as you all know, what is the meaning of the term dye? Dye means color, that changes color when it's put into an acid or base. Okay, now an indicator, it gives different colors in acid and base. Thus what we can say? We can say that an indicator tells us whether the substance we are testing is an acid or base by changing its color. Okay, a substance which contains an acid is said to be acidic, whereas a substance which contains a base is said to be basic. So we can say what? We can say that an indicator tells us whether the substance that we are testing is an acid or basic or neutral by changing its color. These acid base indicators or indicators, they are of two types: natural indicators and synthetic indicators. Okay. Now natural indicators, what are the natural indicators that we are going to learn in this chapter? Litmus, CHINAROSE and turmeric. These are what? These are natural indicators. And phenolphthalein, it is a synthetic indicator, that means it is made in factories. So next we will discuss about what? Next we will discuss about litmus. Litmus as an indicator. Now litmus, it is a natural indicator, point number 1. Now litmus solution, okay, it is actually a purple colored dye which is extracted from a type of a plant called lichen. Now lichen, already you have read in chapter number 1. It is a combination of what? Combination of algae and fungi. It is a kind of symbiotic relationship. Okay, now litmus, it has got a purple color in water, or mauve color you can say in water. Okay, so when litmus solution is neither acid or basic, that means when it is purely it is neutral, it is what the color is purple. Okay, now when this litmus it is added to an acidic solution, it turns red. That means when the solution becomes acidic below 7 pH, when it turns below 7, then it turns what? It turns red. And when the litmus is added to the basic solution, it turns what? It turns blue. Okay, now though the natural color of litmus is purple, it is made into blue litmus and red litmus for the sake of convenience in detecting the color change when an acid or base is added to it. Thus what we can say, litmus is made into two types, one is a blue litmus and another one is a red litmus. Okay, so this litmus, it can be used in the form of solution, okay, or in the form of thin paper strip, okay, of litmus paper, blue litmus paper and red litmus paper. Generally in the laboratory, when we use this litmus indicator, we use the litmus paper strip. Okay, and how does it look? Here you see how does it look. Here you can see the picture of red litmus paper and blue litmus paper. It is made in this kind of paper strip, or sometimes it is made in the solution also. So generally in the laboratory we can use this type of paper strip. Okay, now as I said you that litmus, it is the most common indicator for testing acid and bases in a laboratory. Okay, acid turns blue litmus into red, very important. Acid turns blue litmus into red, and bases it turns the red litmus into blue. Okay, so what we can say, if a drop of a given solution turns blue litmus paper to red, then the given solution will be acidic in nature, or it will be an acid. For example, if we say a drop of lemon juice contains what? Lemon juice contains ascorbic acid or vitamin C. Now it turns what? It turns blue litmus to red. So lemon juice is what? It's acidic in nature. That means if you take a lemon juice in a test tube and then if you dip the blue litmus paper, what will happen? It will change into red in color. And if you put red litmus paper inside the lemon solution, it will remain in red only. Okay, so what we can say, that lemon juice, it contains an acid. Okay, now if a drop of a given solution turns red litmus to blue, then a given solution will be basic in nature. For example, baking soda, it is always available in our kitchen, right? So if we take in a test tube pinch of baking soda, and if we just dissolve it in with water to make a solution of baking soda, and then if we dip red litmus paper, it will turn into blue color. Okay, that means what does it mean? It means that the solution is basic. Okay, it has got pH above 7. Okay, and if we put what? Blue litmus paper, then it will remain blue itself only. Okay, so in order to test a solid substance with a dry litmus paper, first of all you have to make a solution. See, lemon juice, it is itself a liquid, okay, but a baking soda, it's not a liquid. So in order to test it with a litmus paper, first of all you have to make a solution. Okay, so this is all about your litmus paper. So in one word, what we can say? In one word we can say acid turns blue litmus into red. Remember, very important. Okay, that means if you dip a blue litmus paper into an acid, it will turn into red. But if you put red litmus paper into an acidic solution, it will remain in red only. It will not change its color. Okay, similarly bases, it turns what? Red litmus into blue. That means if you put red litmus paper into a basic solution, it will change its color into blue, whereas if you put blue litmus paper into a base, it will remain as blue. This is all about your litmus indicator. Next, what we will discuss? Next we will discuss about CHINAROSE indicator. CHINAROSE, it is a flower. All of you know the scientific name of CHINAROSE is Hibiscus rosa sinensis. Okay, now CHINAROSE indicator, it is a light pink colored solution which is extracted from the red flowers of CHINAROSE plant with water. Now let's discuss. I'm just telling you how this CHINAROSE indicator is prepared. Simply you can prepare this CHINAROSE indicator in your home also, and you can do the test. So how you can make? Collect some petals of CHINAROSE flower and then you place them in a bowl or a beaker. Okay, CHINAROSE flower, you all know which part is known as a petal. Now add some warm water. Okay, now keep this CHINAROSE petal immersed in water for some time till water in a beaker it acquires a light pink color. Okay, so remove the petals after that by filtration. Okay, then what happens, this light pink solution which is obtained, it is a CHINAROSE indicator. Now this CHINAROSE indicator, it can be used for testing acid and base. Now CHINAROSE indicator, what color does it change? As I already said you, what is the natural color of this CHINAROSE indicator? It is pink, light pink. Okay, now if you put acid in a CHINAROSE indicator, it is always in the form of liquid, no paper strip is there. Okay, now if you drop acid, for example if you drop lemon juice, okay, then this CHINAROSE indicator, the color it will change into light pink to deep pink or magenta. Okay, and bases, if you put for example a soap solution, okay, or baking soda which contains base, then what happens, it changes the color into what? Color into green. Here we can see here action of CHINAROSE indicator. Here you can see in the test tube CHINAROSE indicator is there, which is light pink in color, and in the acidic solution, for example in the two beaker, in one beaker suppose lemon juice is there, and suppose in another beaker baking soda solution is there. Okay, now if you put CHINAROSE indicator into the acidic solution, it will change into deep pink in color, whereas if you put it in the basic solution, then it will change into green in color. Okay, this is all about your action of CHINAROSE indicator. The next indicator that we will learn is what? Turmeric indicator. Okay, turmeric powder, it is available in all of our kitchen. Okay, so turmeric, what is the turmeric? It is a bright yellow colored powder obtained from the plant. Okay, in Hindi it is known as what? In Hindi it is known as haldi. Okay, it contains a yellow dye. Okay, now what is its action? Turmeric pepper is yellow in acidic solution. Turmeric pepper itself, it is a yellow color, it remains yellow in the acid solution. Okay, and turmeric pepper, it turns red in a basic solution. Now before I discuss the action of turmeric pepper in details, first of all let me tell you how to make this turmeric pepper. Now this turmeric pepper, this one also you can make it in your home simply. You just listen how to make. Now take a bowl. Okay, now take a tablespoon of turmeric powder, okay, in the bowl. Now then add little water and make a paste. Okay, now deposit this turmeric paste on a blotting paper. If blotting paper is not available, just take a chart paper, cut the chart paper into thin strips, okay, around length, I can say suppose 3 centimeter in length, okay, and 1 centimeter in breadth. Okay, so cut the chart paper, normal white color chart paper, note it down, white color chart paper, cut it like a thin strip, and then you just put that thin strip of the chart paper into that turmeric paste. Make sure the turmeric paste should not be very means the consistency should not be very liquid. It should be like a thick paste. Okay, so this yellow paper thus obtained, it is what? It is a turmeric paper. Okay, now these strips of turmeric paper you can use as what? As indicator. Sometimes if CHINAROSE indicator is not available at home, you can make this turmeric indicator and you can use. Okay, here you see what is its action. As I said you, the turmeric paper, it is yellow in acidic solution and bases turns yellow turmeric paper into red. That means if a drop of a given solution turns yellow turmeric paper into red, okay, then the given solution will be basic in nature. Here you can see the middle one. Okay, here it is turning vermilion or red in color. That means it is what? It is base in nature. That the particular solution or the substance is basic in nature. Okay, so if a drop of baking soda solution is put into the strip of a turmeric paper, or normally if I can say, suppose liquid soap is available at your home, liquid soap or soap, if you make a soap solution and if you just dip that turmeric paper into the soap solution, immediately you will find that it is changing its color from yellow to vermilion or red. Okay, so thus it proves that it contains what? It contains base. Okay, now the yellow turmeric paper, however, it remains yellow on adding acid solution. Understood? Okay, So this is all about your what? Natural indicator. So in natural indicator, what we have learned? We have learned about litmus, we have learned about CHINAROSE, and we have learned about turmeric. Now the next indicator that we will learn is what? Phenolphthalein indicator. Okay, phenolphthalein indicator, it is a synthetic or man-made acid base indicator. Okay, phenolphthalein indicator, it is colorless in acidic solution. Okay, and phenolphthalein indicator, it gives pink color in basic solution. Here you see two conical flask is there. Okay, now here you see phenolphthalein indicator. Here you see one is colorless and another one is what? Another one is, it is pink in color, isn't it? Now see, if we add acid in the solution of a base, neutralization reaction, let me tell you what is neutralization reaction. We will discuss about neutralization reaction later on. Neutralization reaction means when acid reacts with a base, okay, it forms what? It forms salt and water. That is what? That is neutralization reaction. Now generally this phenolphthalein indicator, they are used in the laboratory when we carry out neutralization reaction of an acid or a base. Okay, how? Like if we add acid in a solution of a base, okay, which has been turned pink by phenolphthalein indicator, then the solution, it will change from pink to colorless. Why? Because remember always, acid cancels the effect of base. Okay, now there are some substances which have no effect on any indicator. Such substances are neither acidic nor basic, as I said you. Okay, these type of substances are known as what? Neutral substances. Okay, for example, distilled water, or you can say if you make a solution of sodium chloride, that means a common salt which is available in your home, and if you dip any of the indicator, litmus paper or CHINAROSE indicator or turmeric paper or phenolphthalein, whatever, what you will observe? You will observe no change of color is there. Okay, so these all are what? These all are neither acidic nor basic. They have got pH 7. Remember, they have got pH 7. Okay, and they does not have any change of color on the action of this indicator. Okay, so this is all about your indicator. Okay, a brief explanation of indicator. In our next class, we will discuss in details about the types of acid, what are acids and what are the types of acid. So thank you everyone.